to our 23rd Bioelectrodynamics webinar. This webinar is supported by the International Union of Radio Science, URSI, as well as by Engineering in Medicine and Biology chapter of the Czechoslovakia section of IEEE. Today, I have the pleasure to introduce Hadeel Elayan. She is a PhD candidate at the Electrical and Computer Engineering Department at the University of Toronto. And as you'll see in a moment, her research focuses on stimulating molecular interactions, specifically protein folding using electromagnetic waves, where she proposed that a very original idea that these stimulant-responsive intrabody model, the model she proposed, could be used for controlling the proteins using terahertz. And also, this is perceived by the communication perspective. Hadeel published several papers in high-tier conferences and journals. I would like to highlight that she also received various awards during her PhD, including the 2019-21 Ontario Graduate Scholarships, the PHOTONICS Expansion Grant in 2021, as well as METEX Global Research Award. And she's been named a Rising Star in Electrical Engineering and Computer Engineering Department in the University of Texas at Austin, right? And currently, she's also serving also the community, particularly as a vice chair of IEEE Communication Society chapter in Toronto section. Without further ado, Hadeel, I would like to give word to you, please.
Thank you. Thank you so much for the introduction. And yeah, let us get started. So I'll just share my screen. Okay, I'm so sorry. Let me share. Okay, can you see my screen?
Yes, we do. Perfectly.
Okay, perfect. Okay, so hello everyone, and thank you really, Mikhail, for the invitation. So in today's presentation, I'm going to present our vision, which is basically when wireless communication meets biophysics, we can decode the nanoworld. Specifically, I'm going to focus on the selectivity of protein interactions stimulated by terahertz signals. So our table of content, we're going to start with an introduction, then I'm going to highlight terahertz protein interactions, I'm going to present our system model, some mathematical formulation, I'll share with you some of our results, and finally, I'm going to conclude.
Do you mind going to the full screen if it's possible?
Yeah, sure.
I think it'd be Control L, depends on if you are on Mac or...
Okay, I think, yeah.
Excellent, thank you very much.
You're welcome. So let's start, first of all, by thinking about wireless communication. You know, wireless communication has enabled the connection of billions of people to the internet in order to acquire the benefit of today's digital economy. Telecommunication engineers are working to develop technologies that effectively deliver ultra-high data rates, massive connectivity, and seamless coverage to users. We are all hearing about 5G, beyond 5G technologies, and even 6G, where mobile users communicate with each other or communicate basically with base stations. However, think about this vision. What happens if I move from such big macro scale into a nanoscale? What's going to happen to my wireless communication? How are my standards? How are my protocols basically going to differ? So if I went basically from a macro to a nanoscale, it is important for us as wireless communication engineers to investigate paradigms in order to initiate reliable communication between nanomachines. You might think, why is that? Why not use the conventional methods basically of communicating? Simply because traditional technologies are not suitable due to both the size as well as the power requirements. And hence, we need help here. And this help comes from nanotechnologies, because nanotechnologies, they do provide the engineering community with a new set of tools in order to control matter at the atomic and molecular scale. And think about it this way. At this scale, basically, nanomaterials and nanostructures, they possess new properties that have not been observed at the macroscopic and microscopic levels. And hence, by leveraging and making use of those properties, nano devices can be developed. What are we interested in? Well, we are interested in nanosensors and in nano actuators that can help us monitor and control biological processes at the cellular as well at the subcellular level. But how can those nanomachines basically be constructed? and how they can be made. Well, almost a decade ago, we have this vision which has been proposed by Professor Ian Achildes, Nano Networks being a new frontier in communication. We can think about it as having a top-down approach or a bottom-up approach, i.e., we can make use of the microelectronics, of the microsensors, of the basically CMOS and the semiconductor technologies that we have already developed and see how they can scale down basically to smaller scales into the millimeter or into the micrometer. Or we can also basically look at organelles, we can look at proteins, we can look at the DNA and see basically how do those behave and stem from their behavior and from their functionality. How can nanomachines basically operate? Or we can have a third vision, we can have basically a hybrid approach where we can make use of both electronics and we can make use of the synthetic biology and of the biological components in order to come up basically with those devices. But as long as we have the devices, how can those devices basically communicate with each other? And what is going to be suitable basically in order to establish such sort of communication? And here in the literature, we have two solutions, either molecular communication or electromagnetic nanocommunication. If we take track one, which is the molecular communication track, basically this is the primary method of communication among microorganisms, including the cells in the human body. And molecular communication can be divided into diffusion based communication, advection based communication, or walkaway based communication, but it is solely inspired by basically nature and solely inspired by the intrabody environment. However, we can take also a second track, which is the electromagnetic nanocommunication track. But in order for this track basically to work, I need to have novel nanoantennas and nanotransceivers. So how can those be made? Well, those can be made of graphene, they can be made of metamaterials, they can be made of semiconductor combinations. And those basically devices, in order for us to understand devices at such a small level, basically we need to think about the physics, because the physics is no longer the same physics that we are using. We are now talking about plasma waves. We are now talking about surface plasmon polytrons. So having such combination of devices that are made up of different hybrid materials as well as using plasma physics, this basically points towards how those devices can be constructed. And those devices, they point due to such combination to high frequencies. One of those is the terahertz band as being the frequency range for communication among nanobiosensors. So the terahertz band here, it doesn't come as, being forced that we want to operate at the terahertz band. This comes as a natural conclusion of basically the different combinations that we have in terms of how our transceivers are being constructed. Then comes the question, well, okay, is it suitable to use the terahertz frequency band for intrabody applications in order for us to do, nanobiosensing? Well, yes, because first of all, it is an ionization band where it is biocompatible, and the energy scale of terahertz radiation is within the range of interactions between molecules. We are talking about energies which are on the order of 10 to the power of minus 21 joules. And there is this very comprehensive survey which tackles terahertz technology in biology and medicine that has been published in 2004, and it speaks about and highlights basically how the terahertz band can be used for different biological applications. So now we have this vision. Yes, we are not far from establishing electromagnetic nanocommunications. The purpose of this talk, I will not be highlighting, nano devices or nano antennas, but I would like to point out that if we searched in the literature, the biosensing literature have more than 70,000 publications, which means that basically this field is a growing field. A lot of people are interested in even how to make up those devices. So I'll consider that I have those devices and I'll care about how can I use those devices? How can I establish basically a communication link between those devices in order to suit biological applications? So the objective here, or the objective that we have been working on through the past years, is let us develop theoretical models for intrabody nanonetworks. Through those models, we can capture intrabody interactions. We can understand the electromagnetic propagation mechanism at the terahertz frequency band. And if we looked at this vision, from a body area network perspective, we need to think how to understand the body. I cannot look at the body, just as being stacks of tissues. Well, I need to look at it at the molecular basically level, or even at the cellular level. Perhaps the most accurate way is to study the interaction depending on the wavelength of the interaction. So if you are using wavelengths on the order of nanometers, then you need to ask yourself, well, what can I see through this interaction? If it is on the order of micrometers, well, what will I be targeting? So, and this is here the perspective, is that our interaction should basically depend on the wavelength that we are actually using. And the basis of basically those intrabody models that have been developed since almost, 5 or maybe even 6, 7 years ago, the basis of those is to have a nanotransmitter and to have a nanodetector in an intrabody medium, and try to understand what goes on in terms of their propagation, in terms of the photothermal impact of those devices, both on one another and also on the medium. Yet, when we looked at this basically modeling, we thought that we can take this into a further step. We can do something which is more. And this comes where we have proposed a stimuli responsive paradigm that deploys terahertz waves in order to interact with molecules to control transduction pathways. Think about it this way. I have a nano antenna as being my transmitter, but here, what can be my receiver? Should it be solely only a nanodetector? No, we can use proteins, for example, as being our receivers. And we chose proteins specifically because of the importance of going into protein research and understanding electromagnetic protein interactions. If we do understand proteins, we can understand the protein folding problem. We can further understand structural biology and how we can design drugs. We can further understand the reasons behind neurodegenerative diseases and how a protein misfolds and how proteins also form prion proteins, basically. We can also further understand gene expression. So we think about a protein as being one of the most important natural nano devices that are found in the human body. And by electromagnetically triggering basically protein, we believe that many of the questions that revolve around a protein interactions can basically be answered. And this is going to be the highlight of my talk today. So terahertz protein interactions, our system is basically composed of a nano antenna transmitter and a protein receiver. And why protein in the terahertz band? Because the vibrational modes of proteins are found in the terahertz frequency range. And these modes, according to the literature, they control the protein conformal changes, the ligand binding, and oxidation state. So we think about it this way. By triggering the protein vibrational modes, we can initiate mechanical signaling, and then this is going to result in structural changes and in the activation of biochemical events. So this can be a signal transduction pathway for intrabody, basically a communication. We have our transmitter, we have our receiver, and yet we can here now explore and see what type of information basically I can get. Perhaps I can actuate the protein and send some sort of information, basically. So the work on the literature on terahertz protein interactions is diverse. And here I just chose some of this work. We have the work by Professor Andrea Markels from the University at Buffalo from the physics department. We have several work on the terahertz optical techniques for protein dynamics and characterization. We have also work on terahertz vibration and model analysis in proteins and macromolecular structures. So we can see that the terahertz protein basically field is expanding in the literature with many interesting findings that are basically coming up. We do understand that experimentation is a limitation here, but for now, we are thinking about our model, have a terahertz transmitter, have basically a protein, and see how can this interaction be developed. And the basis of this interaction is going to be as follows. Well, the application of an external electromagnetic field is going to a biological entity, which is the protein in our case, is going to induce a relative redistribution of the internal charges within the molecules with respect to the field lines. And this is going to result in a dielectric response that is is frequency dependent. Subsequently, the nanoantenna electromagnetic fields are going to result in the polarization of protein molecules. And if we examine the protein dielectric response, we can see that dynamical transitions in the protein structure at the terahertz frequency range can be basically attributed to both relaxational responses as well as resonant responses. Relaxational responses arise from the amino acid side chains, while the resonant responses, they stem from the correlated motions of the protein structure itself. And to have a further look into that, if we examine the protein permittivity, well, we can see that the real part of the permittivity, we can divide it into a relaxational response as well as a vibrational response. And this is the vibrational response is this aspect basically that we are interested in. Same thing, if we look at the complex or the imaginary part of the permittivity, we can see that we can divide it into a relaxational response as well as into a vibrational response. So correlated motion basically occurs when many parts of the system are coupled and oscillate simultaneously. And this suggests that, well, if I tuned my nano antenna to the vibrational frequency of the protein, there is a possibility that I can provoke desired functional resonant interactions. And we are see how we basically can take this system model and build from it other stochastic models that capture basically this idea here. So the research question is that, okay, we are saying that I can have a nano antenna protein interaction. Well, the first thing, how to study that, how to evaluate that, how to know that, yes, I'm doing a good job in terms of this type of interaction. So we need a selectivity metric in order to show the capability of the presented system in discriminating the desired response. Yes, I have an nano antenna which is tuned to the resonant frequency of a desired protein, but how can I know how good am I doing in triggering basically that protein? And this is why I need a selectivity metric for that. But before that, how can I develop such a selectivity metric even? Well, I need to bridge the mechanical system of proteins, which can be modeled as harmonic oscillators, and think about what's the probability of protein conformal changes. Well, if I said that I can induce a protein to change its conformation, what's the probability basically of doing so? And what does this probability rely on? And hence here comes the procedure basically of building the model. I can first deploy the Langevin equation in order to capture the protein dynamics. Then we need to find the total energy of the protein, which is stimulated by an external force, which comes from the nano antenna. And then we are going to incorporate the steady state energy of the driven protein into the Boltzmann distribution. So this is going to be basically the three aspects of the model that I'll be looking at closely. So we start and we say that to model the protein dynamics, we use the Langevin stochastic equation under the influence of an external force. So I have the external force, which is coming from my driving nano antenna, but already also I have the stochastic Brownian force, which is acting basically on the protein as seen in equation 3 here, where x of t is the protein coordinate, m is the mass, k is the spring constant, and beta is the damping coefficient. And then we are interested in finding the solution for equation 3 here, which can be decomposed into two parts, one related to the nano antenna external force and the other related to the stochastic force. And in order to solve basically this equation, we have used a Green's function approach to solve the following differential equation as detailed in our paper here. So we can find basically a solution for equation number 3, and the solution is in terms of x external, which is the one due to the driving nano antenna force, and in terms of x zeta of t, which comes from the Brownian stochastic force acting on the protein. Okay, so now what? I have found basically those displacements. I have found what is x external, and I have found what is x zeta of t. But basically, this is not the quantity that I'm interested in. What I'm interested in is being capable of calculating the total energy that has been absorbed by the protein. And when thinking about energy, let's think about it this way. So amino acids that reside in the cytoplasm conducts movements through the rotation of the intracellular churn alongside the molecular interactions among them. ATP usage serves as a biological instigator for the interaction of amino acids. So you can see with such description that we have an active and mass consumption of amino acids, which necessitates the constant flow of high levels of energy, which in absence results in further harm to the cellular functions by disabling the conduction of the protein activities. So being capable of calculating basically the energy that we have in the system, or even the induced energy, this helps us see how can this energy component play a role in terms of how we can control those protein conformal changes. And in order to find the total energy of the protein stimulated by an external force, we have computed both its kinetic and potential energy contributions as follows. So we are looking at energy, we are looking at it at a steady state, and we have divided into kinetic energy as well as potential energy. And since already I know from the previous step, I have solved my differential equation, I know what is my x external, I know what is my x stochastic, I can use those and substitute them in order to find the total steady state energy. The expression for that gave us the following equation here. So it gives us that total steady state energy can be divided actually into a contribution which comes from the terahertz force contribution, which results from the terahertz nano antenna, and we have also the effect, which is the stochastic effect of the medium itself. And we can notice from equation number 5 here that in order for resonances to have an important impact on biological systems, the system must absorb energy in excess of that of the thermal noise. So the system cannot be basically affected by an applied field if its effect is less basically than the corresponding random field. And we can see also from equation number 5 that the contribution of the no thermal noise satisfies basically the equipartition theorem. So now we have moved from the Langevin equation into steady state energy that I'll be making use of in order to calculate what is the probability of inducing a certain conformal change in my protein. But how can I move? And these were the hard parts, or the parts in laying the models, which had to all make sense with each other. How can I move basically from an energy quantity into a probabilistic quantity? And in order to do that, we have deployed the Boltzmann distribution. So basically, energy functions are related to molecular conformation via the Boltzmann distribution. The Boltzmann distribution is known to provide the probability that a system is in a certain state as a function of the state's energy and system temperature. And it is a justifiable selection for modeling the protein states, because proteins act basically as binary switches. So we can think about them as being in a folded state or basically as being in an unfolded state. And this is the link. So to go from basically the Langevin equation, we had to calculate the energy, and then to go into Boltzmann distribution, we had to use this energy and deploy it basically in our protein folding probability equation. So the rate of change of the protein folding probability is given here by equation number 6, where Pf of t is the probability that the protein is in a folded state at time t, Pu of t is basically being in an unfolded state. And we can see that those basically probabilities, they are controlled by rates, Rf and Ru, which are the rate of folding and the rate of unfolding. And from equation number 6, I can move and calculate the steady state probability, which is what is the probability of the protein being in a folded state. And we can see that it relies basically on its rate of folding as well as on the rate of unfolding. It is at the rate of folding where we have incorporated the total steady state energy that we have defined. So we already have a protein which is going from an unfolded to a folded state, but now we are inducing this protein using a terahertz nano antenna. So this rate of folding expression is going to change, where we are going to incorporate basically this total steady state energy in our rate of folding. And this is going to impact basically the probability of being in a folded state, where we can see that now it is no longer only a function of the internal energy of the protein itself, but rather a function of the internal energy of the protein as well as the induced steady state energy. And if we go back to the expression of the induced steady state energy, we can see that how it relies on the different nano antenna basically parameters, including the amplitude of its force, including the resonant frequency itself. So, but okay, like, are we interested in only, targeting a single protein, or are we really interested in targeting a certain population, a certain protein population? And here we can take our model and extend it from being, from being in a single protein state, which we can describe as being a Bernoulli random variable with a probability of success PF. And we can consider now a system composed of n proteins, where the random number of folded proteins basically follow a binomial distribution. And if we assume that we have, well, a large number of proteins, this binomial distribution can be approximated by a normal distribution. So here we can define the protein population as being the number of copies of a protein molecule in a cell, and the number of folded proteins of each population can then be expressed as a normal distribution. And by having this mathematical here assumptions, this allows us now to deal with those proteins as having normal distributions, and we can further and from this step, formulate a lot of stochastic analysis that will enable us to understand better how this nanoantenna protein interaction basically takes place. But as we said, our initial interest is in selectivity of protein interactions. Okay, it's good. We had a model. We started, we found the energy, the total steady state energy of that model. We found what is the probability of the protein basically being in a certain state. But how can we really assess this nanoantenna protein interaction as being, selective enough? Well, when a nanoantenna targets a protein population, our expectation would be, to affect the conformal change of the desired receptor molecule to induce a particular functional response. I don't want to, target a specific protein and I end up folding, let's say I want to target protein A and I end up folding protein B. I don't want to do that. I want to be selective in order to achieve control over the protein network. And if we survey the literature and look at how people define usually selectivity, we can see different, various scenarios. We have in drug discovery, for example. This refers to the experimenter's ability to design selective ligands that act on specific targets. For ion channels, the selectivity features arises at permeable membranes where it allows the passage of some molecules and inhibits the passage of other. Selectivity also appears as a measure of the performance of a radio receiver, where I wanted to respond to a certain radio signal that it is tuned to and basically reject other signals nearby in frequency. So we can see that this concept of being selective, it arises in biology, it arises in different contexts. But our choice of selectivity measure should rely on the interpretation of the metric in terms of our problem, in terms of the analytical properties of our problem, and in terms also of the ease of computation. So we started by, experimenting with different measures that are used in other contexts and applications, mainly from statistics. For example, we looked at the Batcheria distance, we looked at the Kernow distance, we looked at the Kullback-Leibler divergence, but we found that those basically are not suitable for our application since they are not bounded from above. And by looking at the numbers they gave us, well, we couldn't do any connection with how any experimenter can be guided to whether an interaction is basically selective or not. So in order to do that, we said, okay, let's go ahead and let's propose our own selectivity metric, which is given here as an equation number 11. The selectivity metric, basically in the numerator, we have the difference between the probability of the folded state of the desired protein minus the probability of the folded state of the undesired protein. And we can see in the denominator here, we have a maximization function because this is going to ensure the normalization of the metric. And since this developed metric basically relies on the stationary probability difference, then it captures the system mechanical parameters and the amount of energy stored in the protein itself, because basically this is what the probability of being in a folded state is composed of. We know that basically it relies on the energy difference, where the energy itself relies on the mechanical parameters of the protein as well as also the properties of the induced nanoantenna. So we believe that by having this metric here, we can basically capture the selectivity of the system. But still, we need to try this metric in order basically to assess whether it's going to work for us or not. So there is, first of all, we had to explore what are the properties that this metric basically is going to give us. Well, the good thing is that it's bounded between a value of -1 and 1. It has basically a maximum value of 1, and the maximum value of 1, it indicates that both the targeted protein was selected and that the untargeted protein in my system was not selected. And this basically occurs when the probability of folded state is 1 and the probability of folded state of the undesired protein is 0. So, and having basically a value of 1, it indicates this is like the best case scenario that we can get. So it also has a minimum value of -1, and this basically indicates that both the untargeted protein was selected and the targeted protein was not selected. And this is basically going to be the worst case scenario. So if I see a number which is -1, or from 0 to -1. I know that things are ori in my system. I know that the untargeted protein is being selected, and I know that there is something. Maybe my nano antenna is not being tuned to the right frequency. Maybe there are other factors that are basically impacting my nano antenna here. And then we have the value for sure of 0, which is the value of zero crossing, where I'm not selective rather to the targeted or even the untargeted protein population. So I cannot like basically distinguish between both of them. So, and the selectivity matrix is continuous, and it is an increasing function of the probability of a folded state of the desired protein when the probability of folded state of the undesired protein is fixed. So basically, it has some properties which makes it appealing basically in order for it to be used. Now, also, like as I said, I was dealing with singular proteins. Then I have been using protein populations by expanding by functions using the normal distribution analysis. But also when dealing with a protein population, I can also develop a selectivity metric for that. So rather than dealing with the probability of being in a folded state, now I can deal with the mean values of the number of desired and undesired proteins in the population respectively. And the same properties that hold for the previous equation 11 here, it still holds for this equation. So it's just merely expanding my selectivity metric in order to take into account the protein population. And this makes sense because at the end of the day, our adjustment to that is based on the separation of the means of population. And that shall give us also some information about what is happening in our network. Then our final step is that okay, I do have my metric now. So we have developed a selectivity metric. This selectivity metric is based on the mechanical properties of the protein. It is based on how the protein interacts basically with my nano antenna. But can I do something better even? Can I, make sure that I'm triggering a specific protein population in order to activate the desired system response for me? And if we looked back at equation number 5, which was basically the steady state energy equation, we can notice that as experimenters or as people who are working with the model, the nano antenna frequency and the nano antenna force are the only system parameters that we can control. So those are the only system parameters that I have in my hand that okay, I can tune my nano antenna towards it. Other than that, the rest of the parameters, they come either from the protein or they come from the environment itself. So I don't really have control basically over those parameters. So fine, I have my nano antenna force, which I can control. I have my nano antenna frequency also, which can control. So I can lay this basically as an optimization problem. I can say, okay, I have my selectivity metric, and then I can maximize the selectivity metric with respect to the two parameters that I can control, which is basically the nano antenna force and the nano antenna frequency, subject for sure to the constraints of the system. And to solve such optimization problem, we have used the fixed point iteration method, which is a very well known method to solve optimization problems basically. And then out of this optimization problem, what I will get? Well, I'll go to get what is the maximum force and what is the maximum frequency that can I use, and I will guarantee basically that they can give me the maximum selectivity here. And this is basically the joint optimization using fixed point iteration method, where we were optimizing with respect to the force and to the frequency, and until both the frequency and the force itself, they have reached convergence. So, okay, we have, we talked about different layers of the system. We started with our vision of having an intra-body communication system. This vision relies on us having a nano antenna transmitter as well as a protein receiver. Then we said that, okay, how can we develop a metric in order for us to know that, yes, we are targeting the correct protein or the desired protein population. And in order for us to develop that metric, we had to go back to the mechanical properties of the protein itself and then study what sort of total energy is going to be deposited on the protein itself by its interaction with the nano antenna. And from that, we started with the development of our selectivity metric. So in order to test that, we considered several cases of protein populations which are in the vicinity of one another to mimic different intrabody scenarios. Those proteins mainly are that we will be considering are rhodopsin and bacteriorhodopsin. Why is that? Because the aforementioned proteins, they serve as excellent model systems for studying the properties of membrane proteins. This is number one. And also they are well characterized in the literature. There are terahertz spectroscopic analysis that have been done for bacteriorhodopsin and rhodopsin basically at the terahertz frequency range. So in terms of measurements and in terms of values, we have those values from the literature, and we made use of those values in order to study our system. Further on that, we have set our damping value to 0.3 terahertz, and this satisfies basically being in the underdamped regime. I'll be talking about this in towards the end of the presentation, but the presence of underdamped delocalized modes in proteins, they have significant implications for the understanding of the efficiency of ligand binding, protein molecule interactions, as well as biological functions, and this is explained basically in the reference that I'm citing here. Also, for the free energy value, we have set it to 6 kBt, since this value is considered a lower bound for the free energy difference between two states. And unless otherwise stated, we are considering a protein population which is made basically of 1,000 proteins. So then, first of all, how does our protein respond to the nano antenna? So we have on my x-axis the force, on my y-axis I have the mean number of proteins in the folded state. And we can see we have it for protein rhodopsin as well as bacteriorhodopsin. And we can see that, okay, like if I'm using a certain force value, I'm going to activate either one of those proteins. However, if I pushed my force over a certain limit, my both populations are going to be in a folded state in the system. And I don't want that. I want to, activate a certain population, which is either rhodopsin and bacteriorhodopsin, to be in a folded state. And this gives us an indication that our force range shouldn't be an arbitrary range, that there is a specific window. If I exceeded this window, I'll be ending up of activating not the desired protein population. So the protein population basically that I don't want. So I would need to be very careful in understanding what is this frequency window basically for the force that I will be using in order to activate the desired protein. Because if I ended up well activating both desired proteins, then my selectivity is going to be 0, because I cannot distinguish what has been activated from what has not been activated. Then here we have also the mean number of proteins in the folded state basically versus a frequency. And we can notice that each one of those proteins, they have their maximum probability, which then corresponds to also the mean number of proteins in the folded state. It is at its resonant frequency. And this re-emphasizes the importance of basically tuning our antenna to the correct frequency, which is the resonant frequency of the desired protein, or of the desired protein that we want to basically activate. The difference why one of them, like we can see that, for example, the bacteriorhodopsin, its probability of being in a folded state is higher from rhodopsin. This goes back to its mechanical property in terms of its mass and in terms of stiffness. So some proteins are easier to be put in a folded state basically than others, and this for sure is going to reflect on what is the probability of us inducing a conformal change to those proteins. Also, I can extend that basically the selectivity of protein interactions. We can also look at it, for example, if I have 3 proteins. So here in my system, I have introduced 3 proteins at different frequencies, which are 1.1, 1.3, and 1.5. And I can see that all of those proteins basically, because those proteins have the same properties. This is why they have the same mean number of proteins in the folded state. However, what I'm trying here to show that each one of them shows their maximum basically at their resonant frequency, and this should correspond on the selectivity of the protein interaction. So let us measure basically the selectivity of a protein 2 here, and let's look what is going to be the selectivity. Well, in order to understand the selectivity of protein 2, we can see that it achieved basically basically the maximum selectivity at its resonant frequency, which is at the frequency of 1.3 terahertz basically. And we can see at the two other frequencies, which is the frequency of protein 1, the 1.1 terahertz, and protein 3, which was the 1.5 terahertz, I'm achieving a selectivity basically which is a negative value, which means that if I tuned my nano antenna to that value, it's going to give me basically a negative selectivity. And the reason for that, it means that I'm not targeting the correct protein population. So the selectivity basically gives me this mapping of the system. Okay, you have three proteins in three protein populations in your system. You are selecting the second one, which is indicated by basically having the highest selectivity value. But at the end of the day, also it is showing me that the selectivity of protein 1 and the selectivity of protein 3, it is basically negative, which means that I shouldn't basically touch any of those frequencies. Other than that, I will be activating the wrong protein populations. And some of you might say that, okay, although you're dealing with the selectivity of a protein 2, but your selectivity value is 0.8, which is considerably lower. You might have been expected 0.9 or somehow closer to 1, but the issue is that the more proteins I bring into the system, so here I'm distinguishing between three proteins, so the lower the value of the selectivity is going to get. So it becomes more and more challenging if you introduce more proteins in the system, and perhaps maybe even the selectivity metric should be tweaked in order to accommodate for adding more and more number basically of proteins. But the more interesting results come not only from experimenting, with the selectivity metric itself. It basically comes from looking at the results of the joint optimization problem. So the joint optimization problem, we were optimizing our selectivity metric with respect to both the frequency as well as the a nano antenna force. And we can see here, so here what I have in the plot, it is a pseudo color plot which shows the optimal force and frequency for targeting rhodopsin in the presence of bacterial rhodopsin. So I'm targeting rhodopsin, I'm tuning my nano antenna to basically the or my nano the frequency to which rhodopsin is basically sensitive to should be on the order of 1.36 terahertz basically. And it is given here the reaction of basically rhodopsin is given in yellow, while the reaction of bacterial rhodopsin is given in blue here. And this shows basically that if I set my frequency around the resonant frequency 1.36 terahertz, and if I choose a force value of 100 piconewton, I'm really going to get a high selectivity value. So this is the trend that this graph here is trying to show. However, what is going to happen if you chose a frequency which is closer to the frequency of bacterial rhodopsin, basically, then I'm going to activate the wrong protein population, which is shown here in blue. So I have my correct protein, which is basically the rhodopsin. I have my wrong protein, the bacterial rhodopsin, and this optimization method, it shows me, okay, what frequency should I target and at what force. The combination of a frequency and force should guarantee for me what is the maximum sort of selectivity that I can achieve. So can I go ahead and extend this to the three protein example problem that I have shown? Yes. So for three proteins, we can see here that I have now three regions. I have my desired protein, which is shown here in yellow, which is the protein at the 1.3 terahertz. And those are basically my undesired populations, the 1.1 and 1.5 terahertz. And things become more challenging because I will have to be like the range at which this protein is going to be active at in terms of a frequency and in terms of force is going to get basically smaller as I'm adding components to the system. But still, I can say that as long as I set my frequency to a certain value here, which is the intersection between the frequency and force, that is going to give me basically a maximum selectivity, then this is the best that my system can do in terms of how I am interacting with this protein here. And we can notice if we looked at the color bar that the selectivity is lower in comparison to the previous example. Why is that? Because now I'm dealing with distinguishing between three proteins. And as we go like higher in the number of proteins, our selectivity metric is going basically to get lower and lower values. So this is pretty much it in terms of our vision of how we can selectively interact with proteins. And this starts from having a theoretical formulation in terms of how such sort of interaction can be, we can start with. Now, for this type of work, we have considered that my protein is active at a certain frequency. And we do know that a protein have several active modes, and different modes can actually be activated. And this is basically one of the ongoing work that we are trying to do, is that we are trying to see that, okay, if I took the protein structure as is, as having several modes, how can I accommodate for selectivity for that? But it turns out that this is not an easy thing to do, because then the maths, the expansions, and even the interpretation becomes much harder. As we go up in terms of the level. But for now, as a proof of concept, we think that by tuning our nano antenna to the resonant frequency of the protein itself, that there is this opportunity for being basically selective, which reflects back on our nanobiosensing basically applications. Now, what other work are we interested in, or what other work are we also doing in our lab in comparison to the also selectivity work? Well, we can think of our problem as solely from a wireless communication perspective and think about it. What is the capacity of the established link? Capacity is something we communication engineers like to work with a lot, basically. So I have my nano antenna transmitter, right? And I have the protein as a receiver. And when I'm thinking here about a capacity, or when I'm thinking about the problem from an information theoretic perspective, I'm thinking about how putting basically the protein, or how the probability of putting the protein into a folded state can basically be measured. Can I apply Shannon's theorems in order basically to capture such capacity in the system? And what is it going to benefit me, knowing, like if I target the protein using terahertz electromagnetic signals, what will be the capacity basically of the channel? So there is challenges in terms of modeling, but the interesting aspect is then what is capacity in this context, or how we define capacity in this context. And we have already published some work on that, which we call the information rates of controlled protein interactions using terahertz communication. We can also think about the problem from an electromagnetic perspective. What is a suitable propagation model? At the end of the day, in all of those experiments that I have been showing, or the simulations that we have been showing, we have considered that the nano antenna is directly impinging on the protein itself, and there is, like no losses. However, the intrabody medium, it is there is losses which comes from the mobility of the particles, from the absorption of the particles, from the scattering of the particles. So we can think from an EM perspective and see what can be a suitable propagation model. How is the channel really looks like between a transmitter and a protein. Also, one of our ongoing work is to think about this work from a thermodynamic perspective. What is the heating impact? What sort of application I can make use of the heating impact that is occurring in the system. First of all, how to measure it, what sort of application is it related to, is it basically harmful or not? And according to by looking basically at this length scale, which is a nanometric length scale, I would say that models should be tweaked in order to accommodate for that. But the results that we are getting out of that are very interesting. A lot of people think that when dealing with the terahertz frequency band, that there is that there can be those harmful heating effects, but the reality is no. At the end of the day, the terahertz band is an ionization band. If we applied the physics law in a controlled matter, and we know basically what sort of modeling can be used for our system, well, things are on the handled basically aspects. So those are some of the multidisciplinary kind of topics that we are interested in finding answers to. And as a conclusion, so our research basically, it brings together the fields of molecular and electromagnetic nanocommunication, which was the reason why we brought up basically this vision. We wanted a hybrid approach where I can make use of what is already found there in the human body, and also make use of the electromagnetic basically transceivers that are currently being developed. We focused on the link between the nanoantenna and the protein, as well as on the induced responses, and this sheds light on various opportunities where applications can be targeted therapy, biosensing, disease control, and prevention. And we hope that, through this work, we can advance research into the EM-based control of proteins, which we believe will have a great impact on signal transduction. And finally, I know that this research has several gaps, which makes it even more interesting for us. There are more questions to be answered than already those that we worked on. So how to think of developing a complete system, how to understand the time scale of those nano interactions, how to understand the different protein states, how to validate our model through experimental work. So those are all research gaps, but we believe that if we put everything together, it's just like a puzzle. If we have all the elements put together, we really can have a say in terms of how electromagnetic waves can control basically protein interactions, at least from a wireless perspective. We are always very open for collaboration. So if you are interested in any aspect of the project, if you'd like to discuss a specific a certain part which I have talked about, feel free to email me at this email and I'll be happy to answer any of your questions. So thank you very much.
Thank you, Hadeel. Very, very, very interesting, actually. I have many questions. It's great talk. Great talk, actually. I have many questions myself, but let me give first word to our attendees. So I see this question in the chat from Digigo. I'm not sure if they want to ask themselves. So I just make a presenter, Digigo, and let's see if it, or we just can.
I can see the question actually.
Okay.
It says, how does the protein selectivity experiment work roughly, and how does the terahertz field add up? So yeah, so the protein selectivity here, it comes from the fact that we want to see that, okay, we have those, according to the basically the literature, according to the recent experimentation that have been done, protein modes are shown to be active, at least according, for sure, and this depends on the experimentation being done. This depends on, there is a lot of variables to be taken into consideration. But if we took this information as, we have those modes that are active at terahertz, so our interest was how to excite them basically. And in order for us to excite them, we need to come up basically with a selectivity metric. And to do that, we built up our mathematical model. Now, how can this be taken into the next level, which is the experimentation level? This is actually something that we are working on right now. We're trying to collaborate with people who have terahertz experimental test beds, also with biologists, in order for us to know how can we use, what is the most accurate way of using the protein setup, and in order to experiment, at least to start from in vitro study, to see really, are we getting, is there a probability that those protein can change their their conformation if they are being, hit by terahertz signals. So it is the terahertz parts come because we rely on the terahertz part as being our external stimulus. So in order for the local stimulus, which is the mechanical response of the protein, to be generated, I need here an external stimulus, and an external stimulus that I can basically control, which is the terahertz nano antenna here. So it is here where the terahertz basically part add up. Coming up with such, experimental setup won't be easy. However, thinking about it, it needs like having first of all the right equipment, the right setup, and this is what I can say at this stage. But even, the vision for me, exactly how can we transform this and take it into the experimentation level, it's not easy to think about. It needs some, like mechanisms, some inputs, and some collaborative efforts for sure.
Thank you. It was, of course, a broad question and a broad answer. So I have maybe one few more specific. I really like the modeling, especially involving like multiple types of proteins and population of them. And I was mostly curious about well, several things. Let me start one by one. You mentioned that in the rather beginning or first half, that the first two parameters you can control are, you can considered to be controllable externally is a nanoantenna frequency and the force, right? So a force I can understand basically is by controlling the electric field strength you are sending, so basically amplitude or power of the terahertz signal. With the frequency, you mean controlling the frequency of the signal you send, or the natural frequency of the antenna? Because that will be hard to control. It's already set by the
the frequency that we sent. This
okay, yeah, that makes sense. Yeah, that makes sense. Perfect. So in that case, I was wondering, you have this force parameter, which is a very, very crucial parameter also for the optimization. And I'm wondering, how do you couple it to the physical properties? Because obviously it will be proportional. I'm not sure linearly or how with the electric field strength which you apply. But how does it? How do you come up with a particular number? Like, you know, if it's like 100 piconewtons. Did you go like deeper, or you just arbitrarily said, okay, let's consider to be in a piconewton range?
Yeah, this is a very good question. Actually, I realized, so I was looking at the literature in terms of the optical, in terms of the, there is the, now the name is not on the top of my head, but I was looking at the type. There are certain types of analysis that is being done for example, to understand mechanical transduction sort of methods that occur in the human body. And for those, always they were using ranges on the piconewton basically scale.
Yes.
So this is the scale that I decided to start with. I said, okay, since in experimentations they are using those scales, this means, and they are doing, you know, like actual experimentation on those particles. So I said that, okay, as a first start, let this, you know, be the scale that we start with. However, now we can this work because this work was an initial work. However, we took this work also to another level, which is relating the force more reliably to the electric field. What I mean by that is that we can actually compute what is the force depending on what is the maximum electric field that I can give to the system. How do I determine the maximum electric field that I can give to the system? There is boundaries, because certain values of electric fields are going to lead to the proteins being misfolded basically. So if I hit the protein directly, I will misfold them, which means that I will be, you know, like deteriorating the protein functionality from the beginning. So I cannot give this, you know, maximum number. I should certainly be operating at a lower range from that. And we can calculate then the force depending on that values of the electric field, because we know that there is a bar which is a maximum which I cannot hit. But as for those numbers, why they are in the piconewton range, just to re-emphasize, yeah, because depending on the experimentations, this is the range that it has been noticed that it is used, and it is compatible also when I'm talking about, you know, the nanoscale, you know, terahertz waves. I'm not expecting something higher for sure than for that value. Could it be lower? Depending, if we had a look at those results, it seems that having lower frequency ranges basically won't initiate an interaction. So this frequency range is the one that made sense in order for us to invoke and the protein basically. So yeah,
you mean the lower force range? Yeah, sure.
Yes.
Okay, so exactly. I was asking about it because it's exactly, it's biologically relevant force for manipulating the molecules. However, yes, to get such a force by electric field, then you would need to either extremely high electric field or high, and now the question is, what is the coupling parameter there? If it's the charge, if it's the dipole. So I know I'm just wondering about really basics, like if it's the electric field acting on an overall charge, probably it would be a dipole moment of a mode. On dipole, so basically it would be like variation dipole related to the particle vibration mode, right?
Exactly.
And did you okay, so based on this, if you like take just a torque or something like simplified metric to get some, you know, at least some numbers. So did you calculate like how strong the field and the dipole would need to be to get this force? Because for me it's to be like very, very high. I mean, or did you just curious?
It is going to be high. I don't have the number on top of my head, but you are absolutely right. It's going to be like high. It's going to be on the orders of maybe, you know, 10 to the power of, if I'm talking about maybe 10 to the power of 8 coulombs per it's going to be, I don't want to give, you know, just an arbitrary number, but it's going to be a very, very high number. You are absolutely right. Yeah,
because exactly the other questions we were asking ourselves to, you know, what are the, of course in Terahertz it's different, but for pulses, like pulse electric fields which you use, you cannot, there are several limits. Of course there are technological limits, right, to get such a strong pulse actually delivered in the spot. then there are limits for the material. So it would exceed certain threshold, I mean, breakdown of the material, dielectric breakdown, or then, you know, it would be not possible to achieve this field strength to affect the protein. But it's something that's really interesting, so we can maybe chat about it a little later.
Yeah, and there is sorry, yeah, and I do have I do have an understanding also how, you know, the as you mentioned, like how what is the interaction between the electric field and the dipole look like, also like mathematically, because it is interesting, because mathematically this is, you know, something which is established. So then it becomes just kind of comparing the limits, okay, that you have and you want to have on your system with biological limits, and as you mentioned, making sure that you are not breaking basically the physical laws of your system here. Just one thing that I want to add just to this point also, one thing that we are currently looking at is what is the best way also to, you know, to stimulate molecular interactions. You know, like let's say if you have, let's think about it this way: I have a laser, or I have a laser beam, or I have a nano antenna, or whatever my transmitter is. So should I give it like, you know, some sort of a continuous pulse? Should it be, you know, some sort of pulsation? What is the pulse width basically? Should it be nanosecond? Should it be microsecond, millisecond? Some people would argue, okay, you are going to give, you know, short pulses on the nanosecond, but I would tell them, okay, but molecular interactions occur for me at the millisecond. You know, like how can I do this mapping? So this boils down to the same thing. What is the limits, and also what is the sort of pulsation technique that you need to use in order to trigger basically those molecular interactions? So this is also something we are interested in from a thermal perspective, to study, you know, how thermally we can impact our system, but also to see that, okay, maybe, you know, I can get some interesting even characteristics at that length scale. Who knows? Nobody have experimented with that, you know, or nobody have even built theoretical models to see, can I make use of this thing in a positive matter, i.e. can I, you know, invoke some certain stress interactions by having this temperature, but at the same time I'm not harming my system. So you will be standing at a medium sweet spot between, you know, not low intensity but also not very high intensity. So this is one also of the ongoing projects that we are working on.
I see. Yeah, these are exactly excellent questions which we are asking also ourselves. So let me just meanwhile give it, if there anybody having questions, so don't hesitate to ask. I just chatted a bit with the first questioner.
Yes.
But okay, so yeah, and definitely, I mean, it will be depending on typical problem is that if you need to have achieve high force, you need to have high field strength.
Yes.
However, due to the losses you always will have in the system, there'll be a lot of the electric field force basically will induce the heat. So
exactly.
I mean, what we can roughly understand, if you are on these typical scales of, you know, megavolts per meter or maybe even higher, we need to have to affect the proteins. Then if you have a pulse longer than even few nanoseconds, microseconds, there starts to be extensive heating. Of course, depends on the system. If it's nanoscopic system, it's easy. The heat can dissipate. But if it's like more microscopic on a tissue level, that's almost impossible to get high field strength without actually burning and boring the tissue.
Exactly. And this is, and you mentioned one very important thing, which is the difference between, are we doing nanoscale heating or are we doing macro scale heating? Am I interested in micro hyperthermia? Am I interested in hyperthermia? You know, like, and this is, you know, a lot of people would think that even, you know, the question of even what is the best thermal model to be used? You know, this is the first question. Do we go with the heat diffusion equation in order to model that, you know, and solve the differential equations? Would it be enough? Do I need to add anything else? Because the problem is that in the literature, most of the diffusion equation heating, I'm talking here from a thermal perspective, models have been done only for tissues. But what if I'm interested in a section? You know, what if I'm interested in a couple of cells? What if I'm interested in a single protein? So, you know, and then all of this comes. So currently, actually, we are modeling thermal impact at the nanoscale level. For sure, it's still all theory, but maybe this is something that we can, you know, discuss further into. Especially the most important thing in these type of studies is to have a parametric analysis, to know what is our maximum, to know what is our limits, and what values I can, you know, put into my system, just to make sure that, okay, for example, here I mentioned selectivity. I'm looking at it from a positive, you know, perspective, but at the same time, I need to make sure that while doing all that, I'm not causing, you know, any sort of deterioration somewhere, which I'm not, you know, which is not being considered for some reasons, right? So, and this is the issue of having, different approaches. So we are different minds all thinking together. It is the thermodynamic should be put with the biology, should we put people with, electromagnetics and terahertz. And if we looked at this puzzle, as having those pieces together, I think this research field will progress a lot. So yeah.
Yeah, definitely. There are many areas where the questions need to be
Yeah.
answered. If I made one more, actually a few
Yes.
more, but let's go. One other crucial parameter which you mentioned is absolutely, I think, crucial for any selectivity is the damping coefficient. You mentioned in the beginning you always consider larger than 0.3 terahertz damping coefficient. Yes. So probably know by heart, like, do you know to what quality factor it will basically translate? I roughly assume a few units, or what will be quality factor of your antennas? So if it's like 1 terahertz and you have 0.3 terahertz damping, so it'd be something like 3 quality factor of 3, 4, something like that.
Yes, yeah. Now the reason actually, the damping has been an issue for us, for throughout this research, to be honest, because as you know, like if I increase, let's say my damping, if I increase my damping more, I will have, it's like you have no signal. It will be your selectivity is going to simply die out. And the reason behind that is that up until now, we cannot adopt a certain damping model. Like different papers, they give different damping values. So this damping, to be honest, I have chosen just to make sure that we are working in the underdamped regime here, and that damping is not impacting. I supported my the choice of values with some literature work that also consider as proteins being underdamped. But yeah, this is something, and actually one of the great researchers who've I have been recently collaborating with is Dominica. Dominica works on terahertz biomechanics, and he has been invited actually to this lecture series before, and he looks at the problem from a structural perspective, where they try to analyze basically damping. So coming back to your question, even this damping value that we have, it might not be the utmost value. We chose it because by experimenting different set of values, we noticed that if we kept it within the underdamped regime, we will be basically getting good results. How can this be taken into account in a better way? This is something for sure that we have to work on, and we have to make this work even go to the next level. We need to adopt a certain damping model, which we believe just gives me the damping value based on the environment, rather than choosing a damping value which I know is going to keep my system in an underdamped condition. So yeah.
Yeah, it will be fantastic if because damping is a big unknown in this field, because it really depends on particle proteins, and usually people say when you go way below the terahertz, then it starts to be overdamped, because it's related somehow to time scale of the diffusion of molecules, which are the dampers in a way. So it will be actually cool to see you cannot control it as an experimentalist. Well, somehow you could, but actually it will bring a lot of insights if you could show graphs where you have the selectivity or other metrics plotted as a parameter of damping. It will actually bring a lot of insight, because it will also tell us, okay, you cannot operate in certain regime of certain proteins, because this will be giving us overdamped or let's say insufficiently underdamped response, so we cannot actually use it for that. So just saying, it will be actually very, very interesting, because damping is a big question which kills it all if it's too much, and all can bring really intriguing effects if the damping is not high.
And actually, just to back up this point, damping is also one of the now biggest issues that we are having, because we are trying to expand the selectivity metric to take into account 3 and -6 modes, which is basically the number of modes that we are talking about proteins. So now everything for me, it sounds simple. We're dealing with scalars, but then everything has to be like we will be dealing with matrices, and then how do we calculate specifically the damping matrix, which is a correlated matrix? So and then it's interesting if we can find answers to all these questions, but it's also actually very challenging. So but it will be very exciting to have like specifically a damping model which explains what happens at the terahertz, which is backed up for sure with some, sort of experimentations.
Yeah, definitely. I think a lot of insights can be brought from structural perspective, which also Domenico Scavosino is expert in. Also, we
Yes.
are doing this stuff, not published yet, is from a molecular dynamics simulations, which can bring some more insight which you don't usually get from a standard normal mode analysis. And actually, if you go to the literature, maybe you had already found that, just maybe highlighting it might be interesting for you. There are very nice works which show from molecular dynamics simulations that if you apply frequencies on the subterahertz, the terahertz range, there are effects on statistics of the behavior of the water. For example, even the Digigo who's asking the question is author of some of these works, and maybe hopefully we'll have him for one of our next webinars. And also Niall English from Ireland is one of the experts who actually was doing MD simulations since a long time on the microwave electric field effects on the proteins. And there they actually explored what are necessary field strength to see any effects on the time scale of nanoseconds. So this brings me to one more, I think, conceptual point, I think, which is always I like to raise. And it's always like, if you want to see the effects of electric fields, either you act strong enough for a short time or long enough for a weaker field. So because there is some scaling probably, and we need to find a sweet point, like what is the appropriate length. Okay, let's say we don't want to heat up the system, but we want to probably act long enough to bias the statistics so we can see the effects on the long run. And for protein folding, well, this might be important because it can take milliseconds, right, or other phenomena which are protein time scales. So it would be actually interesting to see also. We work with the total energies, I think, which is a very smart approach. My question was here, like, if you have a concept like, if to take into account not only the force, but also how long the force acts. So basically it's kind of integral, which you would be getting energy in the end. That's true. But yeah, I'm just curious. Yeah, there's another thing, like, which if you consider, the length of the effect, you need to length of the exposure, let's say, to electric field to see the effects.
No, like in the selectivity work, no, but this is actually, it would be very interesting to see. I didn't think about it from a, spatial temporal perspective, which is having it versus time, but it will be really very interesting. We didn't see that for the selectivity effect, but right now the work that I'm doing on thermal analysis, for sure, time is an integral factor for us, because we want to look at the transient heating, but at the same time, we want to look at steady state heating. So, and here time, becomes really a critical factor, because as you mentioned, maybe after doing the thermal model we can connect those together, thermal with the selectivity. But yeah, time is fundamental. Acting in a steady state gave us this was a smart way of us thinking about the analysis, that okay, we look at energy, we look at it at steady state. So this made us okay for this model. Yeah, it works fine, but definitely looking at time is something crucial. Yeah, so it's some of our ongoing future directions to incorporate time.
Yeah, I was also curious, like in in biological settings, it seems to be almost impossible, and we discussed that with Andrea Markels, who was the webinar some time ago, to actually see selectivity on a if you have thousands of proteins in this one volume. So where we are going right now is more like having isolated systems where you really know, like with one protein, actually to see any effect of that, of the subterahertz pulse electric field on that. And that's something which could be, I think, a connecting point we'll be also interested in.
Exactly, because at the end of the day, despite the fact that yes, for sure, if we had and this show was very even vivid for us, even if expanding it to just moving to 3 populations or even 4 protein populations, so it was vivid for us that it becomes much harder. But as you mentioned, at least understanding what happens at the single protein level is important, because this shows us that yes, we can induce some sort of responses which we can make use of, or it means that no, like everything is just washed out with damping or washed out thermally and there is nothing. So until now there is diverse work in the literature, but there isn't, for this very simple question, there isn't this a simple answer. Is it a yes or is it a no? There is all of those works, all those experiments. It will be very good if we reached a point that yes, we are sure, we don't mind that if we have if the system, for sure, like if we're dealing with multiple proteins, it becomes harder, but at least at the single level, or at a couple of protein level, or at a small population scale, to see something. And we know that this response is coming from the protein, because sometimes you induce stress to your system, you give it some electromagnetic signal, and you see a response. But how do you know that this response is coming from the the intended interaction? So it's this link which is also missing in the picture itself. Work in the literature is promising. I have been finding papers since 1998, which are talking about the microwave interactions with proteins, and they have exactly a very similar vision into whether these effects are thermal or athermal, whether a protein is going to be stimulated or not. But as I said, a clear answer to this, I think this is what is needed. So yeah.
Completely agree.
Yeah.
Okay, great. Fantastic. So I don't see any more questions now from the audience, but I think it was really great. So yeah, what I'll do now, I'll just stop the recording, and then you can chat a little bit more. Or somebody maybe can ask visual recording if somebody's shy to do so. So before stopping it, I would like to thank to all our attendees and our speaker, and hope to see you on the next webinar. I'm stopping the recording now.